Hello class, my name is Vivian, and for my final microbial project I will talk about Streptococcus pyogenes, Candida albicans, and Ebola virus. Streptococcus pyogenes is a gram-positive bacteria, non-model, facultative anaerobe, catalyze negative, with a round to ovoid shape that grows in a long arranged chain. Its metabolism is fermentative, reproducing by binary fusion. Like all gram-positive bacteria, its cell wall is thick that stains purple in gram technique. It is a mesophilic bacteria and can be considered an opportunistic pathogen. I think this slide represents how people know Streptococcus pyogenes, because the reservoir of this pathogen is the gastrointestinal human tract. In a long life, people can feel some symptoms of this cocci, as a strep throat or pharyngitis. These two diseases are non-invasive, but there are invasive diseases caused by Streptococcus pyogenes, like impetigo, scarlet fever, pneumonia, and even endocarditis. It is important to say that when pyogen is not well treated, it can travel through the bloodstream, affecting other organs, as the heart or the lungs, for example. There are 4 different types of Streptococcus bacteria: type A, type B, type C, and D. S. pyogenes belongs to group A, beta hemolytic, that can cause a range of diseases. The treatment more common is drugs that target proteins that catalyze peptidoglycan synthesis. Penicillin is usual treatment against strep diseases, but there are alternative drugs. It depends on the organ or on the severity of the disease. I put here in the slide some medicines that can be alternative to penicillin. These are the main Streptococcus pyogenes virulence factors, but M protein is considered its main virulence factor. What is M protein? M protein is a virulence factor that can be produced by certain species of Streptococcus. Viruses, parasites, and bacteria are covered in protein and sugar molecules that help them gain entry into a host by counteracting the host defenses. There are efforts to produce a vaccine for S. pyogenes, but somehow the ones studied until now bring concern about antigens that may contain autoimmune epitopes that could trigger the disease that the vaccine is designed to fight against. So, no vaccines for S. pyogenes until now. Candida albicans, an yeast form unicellular reproduced by budding. Single budding of the cells may be seen. Both yeast and pseudohyphae are gram-positive, and this is a unique characteristic of this microbe. It can switch between different phenotypes. This can happen multiple times because Candida albicans is a polymorphic fungus. It is encapsulated and diploid, also form two hyphae, can form biofilms, but Candida albicans' normal condition is yeast. Candida albicans is a fungus naturally present in the body without causing infection or any symptom. It lives in balance with the host. Usually, Candida albicans can be found in various parts of the body, being more frequent in the vaginal mucosa of women, oral cavity, gastrointestinal, and urinary tract. Only when there is a change in the balance of the organism, Candida albicans can proliferate and cause some disease. Symptoms of Candida albicans infection vary depending on where the fungus developed. In the case of oral candidiasis, plaques and white dots usually appears in the mouth, cheeks, tongue, and throat. This type of infection is more common in newborns whose mother had vaginal candidiasis during pregnancy. In the case of vaginal candidiasis, symptoms can be uncomfortable due to the burning and itching that this pathogen normally causes. The diagnosis of Candida albicans infection is made initially through the assessment of symptoms, but laboratory tests are necessary to confirm the diagnosis. The main virulence factors that promote fungal colonization of the host include, above all these ones, the ability to adherence to host cells and resist physical removal, invasion of the host cells, metabolic flexibility, competition for nutrients, resistance to innate immune defense, and evasion of adaptive immune defenses. Mild or moderate genital candida infections can be treated with a short course of an over-the-counter or a prescription of an antifungal cream, pill, or suppository. It can also be prescribed a single dose of an oral antifungal medication, such as fluconazole. Unfortunately, there are no immunotherapies or vaccines approved for the treatment or prevention of fungal infections. Ebola virus. Ebola is considered a zoonosis, meaning that the virus is present in animals and is transmitted to humans. Filoviruses are single linear non-segmented RNA viruses. They are polymorphic and have different lengths. There are four known strains of Ebola: Ebola Zaire, Ebola Sudan, Ebola Cote d'Ivoire, and Ebola Ranston. Ebola virus is a highly virulent pathogen. The Ebola virus is the prototype of a viral infection that causes hemorrhagic fever. Ebola is one of the most virulent human pathogens. Leads to severe disease and has a high lethality. This high mortality, combined with the lack of treatment and vaccination options, make Ebola a huge public health concern. One of the main reasons that the Ebola virus is so deadly is because it disrupts the body's immune response to the infection. Ebola is highly contagious by direct contact with other body fluids, such as infected blood, secretions like vomit, diarrhea, urine, breast milk, sweat, semen, and other body fluids from dead or living infected. Not only health workers but all people who need to approach patients with a confirmed case of Ebola or even suspicion of the disease are required to wear protective equipment that cover the body from head to toe. And when removing the equipment, must take care. Hygiene and personal care must be redoubled in areas where Ebola may be infected. The only therapeutic resource against Ebola infection is to offer supportive measures, such as electrolyte replacements, hydration, control of blood pressure and blood oxygenation levels, in addition to the treatment of infections complications that may arise. In fact, there is no cure until now for Ebola, but there is hope. In December 2019, the US Food and Drug Administration approved Inmazeb, a mixture of three monoclonal antibodies, as the first FDA-approved treatment for SARS Ebola virus infection for adults and pediatric patients. Ebola is still limited to parts of Africa,